Good evening, everyone. What I'd like to do here in the next 20, 30 minutes is I'd go over a few items. First of all, just talk a little bit about genetic selection and how it works and what progress we're making, and especially in light of the ability of genomic testing over the past 9 or 10 years. And then get, I think, into what's really the meat of it, which will be how does genetic potential interact with nutrition and management, and then talk about our feed efficiency project with which Dr. Santos and I are both part of, and then try to throw out some things that we might chew on in the Q&A session. Any genetics presentation has to have this equation in there, or at least we usually do. And this is really a report card is kind of what I want to do here. Selection intensity is pretty simple in dairy cattle with the great work that our reprophysiologists have done over the last 50 or 75 years, where we need very few males, obviously, as parents, but now very few females as well. And we can get really massive selection intensity, top 10th of 1% type of thing for breeding animals, and propagate them really widely. The other one that we're doing great on is this genetic variation. Almost any trait that we care about seems to have plenty of genetic variation to make progress. That's not really a big issue. The two that are a bit of a struggle are the accuracy and the generation interval. For accuracy, we do a great job for traits like milk production, where we have lots of data and the data are of good quality. We have other traits, say like female fertility, where we have lots of data, but the quality is not great, and we have kind of rough measures that we work with, like conception rate or days open, but we don't really have detailed physiology data on a lot of on large populations. And then we have the really hard traits, things like feed efficiency, where it costs a ton of money to measure the trait and to do it well. And there we really have low accuracy at this point, but we're trying, right? And then generation interval used to be an issue, but I'll argue now that with genomic selection, that's really not anymore an issue for cattle. Just a few stats. First of all, I don't expect you to read this giant table, though you can go to the CDC website if you'd like to. Over 4 million animals, dairy cattle, have been tested in the past decade since the DNA microarrays became really widely available, and also since we drove the cost down to something that was affordable. And that equates to about 60,000 calves a month. And really every genetically relevant animal for breeding stock is tested. You would hardly be able to, we certainly can't buy semen from a bull that's not tested. If you go to an auction or sale, you probably aren't going to find an animal that's not genetically tested. But really large presence on the commercial farms as well, with 700,000-800,000 calves being tested every year. So many herds now have multiple generations where all animals have been tested, and it runs about, there are many different options, but typically what most people do is about $40 an animal. Okay. I talked a little bit about this generation interval, and this is a paper in the PNAS Proceedings in the National Academy that was published now 4 years ago, and the trends continue to go downward, at least in this red line up here. But what you'll see is that the parents of bulls now that go into the AI company are always first calf, heifer, and a young genomic bull. That's almost never the case that it's an older animal. And the parents of replacement heifers are much younger as well. The age generation interval there on both the male and female side is now in that 3 years something range, and so we've driven that down too. And that matters because it takes a long time to make genetic progress. This is why geneticists in genetics departments don't use cows for their long-term projects. They use fruit flies and mice and stuff like that, because otherwise they would make no progress in their lifetimes, or very little. The trends I show here, just one trait for milk yield breeding value. This is U.S. Holsteins, and you can see one nice long upward trend going back 50, 60 years, but it's accelerating here at the end. The bulls are always ahead of the cows because they're more highly selected. There are fewer of the bulls make it into this graph, right? Most of them go to beef. But we see 126, 156, 209, 250, so we're accelerating. and potential is great. And then what we need is folks like Dr. Santos to help us actually realize that potential, because geneticists are good with numbers, not so good with rations, right? I'll show a couple other sort of features, and I'll mention that many universities did these long-term selection projects initiated in the 1950s and 1960s, and I'll just point out results of two of them here. One is, and probably the two most successful, which were not here in Madison. The Iowa State one had selection of high versus average production level sires, and then did this for many, many generations. And one of the things they found was this: yes, they made more milk, but then they also had this undesirable correlated response in health traits. So the cows that made more milk also had more health problems and higher health costs. And so sometimes you get what you want, and sometimes you get what you want plus some other stuff too. And I show down in the lower right the graph in U.S. Holsteins for daughter preg rate, which is a female fertility measure, and you can see that it was getting worse and worse and worse for about 40 or 50 years there, until the last 10 to 15, when we started focusing on leveling that off and stopping that deterioration. And so we need to always think about that, and how are we changing the cow intentionally, and then how are we changing her unintentionally through that selection process. The other one I'll mention that's more relevant to this discussion is kind of a really clever one at University of Minnesota. It was set up by Charlie Young many, many years ago. And in the 1960s, or early 1960s, they started saving, using just repeatedly semen from bulls of that era. And so you really had a static control line that wasn't getting dragged along, making genetic progress by accident, like the control line in Iowa State. And so you had a control line there that did not change genetically by definition. And then the selection line that improved quite dramatically over time, like the rest of the breed. But what was interesting is the control line didn't improve phenotypically either. And you would say, wow, that's because all nutritionists and other dairy management type folks were on vacation in the 1960s, 70s, and they all didn't come back maybe until the late 80s. I doubt that's true. And I think what really happens there is there's no genetic potential, and so you can feed these cows, you can improve their cow comfort, and nothing really happens. And you can imagine, for example, if you took a Hereford cow and started feeding her, large quantities of nice dairy cow TMR, that you're not going to get all that much milk, right? She probably won't eat it at all, and if she does, she'll just get fat, right? So these things go hand in hand, and I really love this lower one from Australia to kind of demonstrate this principle. And so you've got 3 blocks there, and you can go to the paper if you like, and see all the details of Fulkerson's, but you've got breeding value on the x-axis, and then on the y-axis or vertical axis, you've got actual performance, but then you've got it at 3 levels of concentrate. And what's really important here is, as the level of concentrate feeding, these are cows that are primarily pasture with supplemental concentrate, and as the level of concentrate feeding goes up, the slope of the line gets steeper and steeper and steeper. And that's demonstrating exactly this point, that genetics and management have to go hand in hand, and particularly genetics and nutrition. You can't just breed cows better and not feed them, but you also can't just feed them better without breeding. And in order to realize that potential, you've got to manage for it. So setting that table, I'll talk now a little bit about our feed efficiency project, and then Jose will talk more about some of the work they're doing at Florida later. Mike Vandahar has been the leader of this big project now for 8-9 years, something like that, at Michigan State, and a bunch of universities are involved, and I'll show a few of them later. But the point of this slide, and you guys understand this better, the guys and gals understand this better than I do, but we can improve performance and we get more efficient, but at some point those returns are diminishing. And why are they diminishing? Well, because rate of passage, things like that. And so if you look at the blue line of the NRC, the old NRC 2001, you actually see that once you get up to like 5 multiples of maintenance, you're getting less efficient. I think that's just an artifact that they had very few cows. These would be data from the 1980s and early mid 90s, and there weren't that many cows at university farms in experiments that were 5x maintenance. And so you fit a quadratic or a nonlinear curve to it, and at the tails of that curve, it kind of goes crazy, right? So I don't really think that's what's going to happen. The green line would say there's no digestibility loss. So that's probably not right either. And our data shows something kind of in between, but you do see that kind of leveling off. Well, why is that important? Well, because that tells us that if we want to improve, I say feed efficiency, but energy utilization efficiency, uh, we need to actually measure individual cow intakes. We can't just select for high production and hope that they get more efficient too. We have to figure out which cows are biologically more efficient. So if one wants to do that, and one wanted to do that before, say, 2009 or 2010, it was almost impossible. And some people like Roel Veerkamp and the Netherlands and others did a lot of good work back in the 80s and 90s on feed efficiency and kind of gave up, because in a progeny testing system, which is our old world, you would have to measure every daughter of every bull. So 1,500-ish bulls a year in the United States alone. You'd have to measure every daughter, say 100 daughters per bull. So you'd have to measure feed intake, say 6 weeks mid-lactation for 150,000 cows a year, which if you could do it, which you couldn't because such facilities don't exist, but if you could, you'd spend like $75 million a year or $50,000 per bull just to get a breeding value estimate for feed efficiency. Now with genomics, we can develop a reference population. I'll talk a little bit more about what that is exactly later if you're not sure, but build that reference population, spend a bunch of money up front. This is what we're doing right now, and then add to it over time, and you can decrease the cost by a couple orders of magnitude. So that's why we're coming back and revisiting this now, that in conjunction with the last slide about multiples of maintenance. 4 steps of genomic selection. 3 are really sort of mechanical. 1 is a lot of work. So the first one is building this reference population, getting a few thousand bulls at least that have lots of daughters with performance data, and the bulls have been DNA tested, or tens of thousands of cows with DNA test and performance data for the trait you care about. Once you build that, then you can do the rest. The mechanical parts, estimate the SNP effects or the breeding values, which is the sum of those across the genome, test some calves and make decisions. But this is really the big one, and it's the hard one, is this reference population. It's great for things that are measured by DHI or type traits measured by breed associations routinely, but for traits like feed efficiency, or, we did some work last year with ultrasound diagnosis of lung respiratory disease in calves. I mean, it's hard to build those, but it can be done. This feed efficiency project, first we had a USDA project, same universities plus a couple more, and then we kind of zeroed in on these 4 plus USDA in the next project, which is funded by the Foundation for Food and Ag Research and the Council on Dairy Cattle Breeding, that's matching funds. And we measure a little over 800 cows a year at the different university farms, as many as we can, given our facilities, Kalen Gates or Grow Safe or Incentech in our case, or in some cases it's just by Waybacks. But the idea is to build this reference population, get more reliable genomic predictions for feed efficiency, and I'll talk about what that looks like in terms of the trait in a moment, and then develop some other sensor measures, measure heat loss, measure various blood parameters, use cameras, do anything we can to figure out how to predict dry matter intake as accurately as possible. So maybe we can then at some point do this on commercial farms that don't have to buy a Grow Safe or an Incentech system that would be not affordable. And so the trait that we're looking at, and a lot of the early work was done in beef cattle. A lot of the work in chickens and pigs tends to be more looking at ratio traits, but in beef cattle, it's more residual feed intake has been the norm. So how much did the animal eat versus how much do you expect her to eat is sort of one way to do it. You can calculate based on book values of requirements, or you can do it based on contemporary comparisons. So how much did she eat relative to her cohort after you adjust for metabolic body weight, energy content in milk, secreted body weight change, and that sort of thing. And you can do this, I forgot to mention this morning, this is in kilos, but we can do it in megacals and usually do, honestly. But what you want to do is find cows that are producing at a high level and also eating a little bit less than they need, versus cows that are eating more than they need or more than their contemporaries do. to get the same output. And we really, often get the question about, well, do we really want a cow that doesn't give any milk but doesn't eat much? No, we don't, right? We want both. So we want high performance, plus we want it done efficiently. Okay. And there's some challenges with this trait that we'll get into later. So yes, well, we start with the gross energy. We got what's used as to get to net energy. There's the RFI part and then dilution of maintenance, and you guys understand all that better than I do as a geneticist. But one of the interesting things we found as well in our data, and others have started to see this earlier with smaller data sets, is you've got the maintenance requirement going up. And so the latest research, Templeman is about 0.11, so that's about a third higher maintenance cost than it used to be. So why is that? Well, we're changing the cow, we're changing the cow's environment, and we wouldn't necessarily expect it to stay the same, but it's actually a pretty big change and a significant change that I think we need to talk about. I'll talk a little bit about, so we're probably going to get feed efficiency breeding values, the preliminary ones released in December, and then later in the winter or early spring, we'll get everybody in the US will get breeding values for feed efficiency for their cows and bulls. And this would be done as feed saved. And this graph in the upper right is from Jenny Price and her colleagues in Australia, and they kind of pioneered this, and I really like the idea. So you take residual feed intake. Did she eat more than she should have based on her size and her milk output and milk energy output? And then you also add in the excess maintenance costs, because RFI doesn't care if you're big or small. We take out the metabolic body weight part, right? But we do need to penalize you for just costing too much because you're too damn big, right? And so if you add those two together, then you get feed save. But what she's shown is that cows are getting bigger in Australia, so the maintenance costs have been going up. The RFI has been going up a little bit as well, and so feed saved actually is going in the wrong direction, and so they want to reverse that trend and start to at least stabilize that, if not improve it. Genetic standard deviation is shown here. It'd be bigger in our case because of scaling. Our cows are eating more, producing more, so the genetic standard deviation is quite a bit higher. And then they also looked at another study as what is efficiency as a heifer, and then is that the same trait as efficiency in a lactating cow? And there is a relationship. So if you take the highest and lowest heifers, 100 per group in this study of Davis, and you look at what happened to them, and that's RFI as a growing heifer. So all that's in the equation is the growth, body size and growth rate. What happens when you throw in the lactation part? And they did still see a difference, right? It's smaller. Okay, the correlation I think between the two was around 0.3, but it's at least good news that the correlation seems to be in the right direction, and it does sort of diminish over time. If you were looking at heifer versus third lactation cow, it's closer to 0, but at least it's not in the wrong direction. And most of our work's done in lactating cows, but there are folks, especially ST Genetics, who's done a lot of work in growing dairy heifers. What is it biologically? This is in beef cattle. I think we're trying to understand more about what it is in dairy cattle with all of these add-on experiments that we're doing along with our feed efficiency data collection. We're overlaying nutrition trials and physiology experiments and data collection of blood and tissue samples and measuring kind of anything we can, behavioral data. We see digestibility as part of that. Body composition, that's something we haven't really addressed. When you're just looking at metabolic body weight, you don't care if she's, for lack of a better word, tall and skinny or short and fat, but that matters, right? Physical activity matters, heat loss matters, and then this protein turnover seems to be a big part, and so we need to look at how we address that. And then there's an unknown part, and some of that unknown honestly is error, right? Because RFI is the leftover after you've accounted for all of the energy sinks that you know about and you believe in, right? And some of those might be important. And if immunity or something is an energy sink and we ignore it, then we're going to create ourselves a problem, right? Because we're going to select cows that are not optimum for those traits. I'm not going to go through all these other species. Well, there's cattle too, but it's mostly pigs and chickens and mice. But you can see on the slide in these selection line experiments what changed. Body composition changed, although not always in the same direction. Protein turnover always decreased in the selection line. Mitochondrial function changed. Digestibility either didn't change or improved. Physical activity generally, decreased, right? So that's some of the things that are happening within the animal when we're just selecting for what we see on the outside. And then I threw this in kind of for fun because a few years ago I did a talk, and a couple of years ago, and Lloyd Holterman said, well, here, look at this data that I've got from our herd. And it's an association study, of course, not even a study, right? It's just an association. But, 9 years difference here from when he started these calculations, and he monitors every year the health costs. How many dollars is he's spending on health costs, veterinary bills, that sort of thing per hundredweight of milk? It's gone down from $1 to 31 cents at the same time as feed efficiency has gone up. And on a farm of 1,100 cows, that's 325 grand. So nothing to sneeze at there. So, and I think Jose may mention this as well later on, this relationship between healthy cows and feed efficiency. That's good as well. We don't want it to be the other way around. I put lots of actions on here that you can take now that we have this sort of, we've gone from when I was growing up, when you ignored the young stock and then revisited them 2 years later out somewhere on a pasture, to now we can do active inventory management. We can create the inventory we want, and we can manage it as we choose. As genomic testing started, we started with this culling the low end of animals, and then we started with sex semen, and then with IVF, and now beef semen, and in just a 10-year period, we've evolved to a point where we've got an enormous quantity, hundreds and hundreds of thousands of dairy cows being bred to beef bulls because we have sex semen for the top end, or sex semen for the young animals, beef for the old, and we can really do some things with the production system that we wouldn't have even dreamed about a decade earlier. Okay, and that's why we have to take all these tools and put them in a box and shuffle them around, and then or shake them up a little bit, and then dream up some new scenarios that we can take a look at. And I'll talk about a couple of others. Another thing we can do, and we've worked on, others will work on, this is the cow version of personalized medicine. In this case, we were predicting blood BHB in the first, between 5 and 18 days in milk, I think. And we can do that with the environmental variables, the herd variables, if you will, or demographics, and we add in the genomics, and we can predict which cows are going to be above that 1.2 threshold and which ones are not. And there's a long ways to go on this, but I think it's a really fruitful area going forward, especially as we get the ability to integrate sensor data and video and all those sorts of things to identify cows that might have problems. I like this slide just simply, well, 2 reasons. Mostly because this enormous cow that is apparently 8 feet tall and the woman that's maybe 4 feet tall, but it demonstrates that we as geneticists are always thinking about that extreme, pick that top 1/10 of 1% and try and make more like that. But uniformity has real value, and we have not in selection programs focused on uniformity. And I think we could, well, I know we can do that. It's easy to math, but we haven't chosen to do that. But that is something that we certainly should consider going forward, because we aren't managing individuals, or managing groups, and uniformity is valuable. Okay. Lots of data on this slide, but the point is, if you take, again, this is the sort of, this is UW-Madison herd data from a few years ago when I made this, but it's the equivalent of that Australian study I showed you earlier. You've got the genomic breeding value, or in this case, the predicted transmitting ability, which is half the breeding value, as a calf. So the predicted transmitting ability from the DNA test closest to 12 months of age for an animal, and then her actual first lactation. It's actually ME first lactation, ME of her first lactation yield, if that makes sense, sorry. And the slope of this line, this is the point, it varies a lot from herd to herd. Now you've got the sick animals at the bottom, that makes sense, right? But and the more sick animals you have, the more that's going to pull the slope of the line down. But you've got herds that are really have a steep curve, and they're getting every ounce of genetic potential out of those animals, and you've got other herds that just frankly aren't. And I think there's a lot of room on a herd basis, or a pen basis, or a subset basis first lactation cows or third lactation cows, open cows, pregnant cows, whatever, for using genomic potential as a management diagnostic, because we know now so accurately from the DNA test what's the potential of that animal, and now we can look at a management intervention and see, does she respond or not. Okay. And then lastly, I threw this in here just for fun, kind of, but you think about that normal bell-shaped curve and all the different options we have for our replacement heifers. We can think about the cows too, and what do we do differently? Some 15 years ago, when I was early in the extension specialist career, I put together this picture of Kepke's granny cow that had 430,000 pounds of milk or something like that, 16 calves in 17 years. That's kind of the perfect cow that had no events in dairy comp. She just did everything, or very few events, because she just did everything quietly, and all of a sudden you realized, wow, but is that the goal? Do we want to keep every cow as long as we can? Probably not, right? And we can think about strategies now, for example, getting all our replacement heifers from our first lactation animals, or from our yearling heifers in first lactation, and then everybody who's over 2 years old gets beef semen. We can do that, and then we can keep some of those cows around, the older cows, but we never have to get genetics from them, because we want to get the genetics from the younger ones that have better potential. The older ones we just breed to keep lactating. We can do that sort of thing, or we can even think about scheduled replacement. And I put the example here of a rental car company. Like a bad motto for a rental car company, I think, if I were a marketing person, would be, we drive our cars until you're stranded by the road, right? And if we think about how to optimize our system in terms of not just performance of the dairy cow as a dairy cow, but performance of the system in terms of the dairy part of it, the beef production side product of it, the animal welfare part of it, the consumer perception, and all those things. I mean, maybe we should imagine a scenario, or at least evaluate economically and socially a scenario where you get 2 calves, you don't breed her back in second lactation, but at the end of that lactation, you move her to a feedlot. There are many pitfalls with it, but that's just an example of the broader type of thinking we need to do, because we have so many tools now that we can do this quite easily if we choose to. I'll stop there. I got a few take-home messages. The genomic testing at kind of the 10-year mark now has had a massive impact. If we measure the right traits and do it correctly, we can change them very, very quickly. We can't have genetic improvement without nutritionists, and so like it or not, Jose, we're stuck with each other. If we want to make potential and then turn that potential into for performance. Feed efficiency, we're spending a ton of money getting a ton of data on the genetics side, also on the biology side, trying to learn as much as we can about relationships, what happens when you change rations, and more fiber and less fiber, and higher protein, less protein, and all those sorts of things. Are there negative health implications? And you'll talk about some of those in a minute, but we need to learn more, and then we can put all these tools in our toolbox, and then put our thinking caps on and try and come up with some new strategies. I will stop there and turn the reins over to Dr. Santos.
I'm going to start my presentation with a question that I posed to a group of nutritionists in different parts of the country. So I sent a question and I asked them to identify what dietary interventions will take your best clients, your best herds to the next level of production. And the thermometer here was energy corrected milk. And I gave them 10 options, and I asked them to select the top 3. And then I summarized the top 3, which I'll show you later. So the possible choices were improved forage NDF digestion, be able to feed higher forage diets, increase the supply of metabolizable protein, increase duodenal delivery of essential amino acids, deliver essential fatty acids to the small intestine in a more precise manner, increase feed intake in early lactation, feed diets that prevent early lactation disease, feed diets according to level of production, prevent rumen acidosis, and lastly, feed diets that reduce inflammation. So when I sent this by email and I asked them to pick 3, different people pick different choices, but the top 3 choices were the ones that are listed here. So improve forage NDF digestion, prevent early lactation disease, and increase feed intake in early lactation. So I'm going to spend a little bit of time in each of those, and I'm going to try to identify whether what the nutritionists identified as being bottlenecks for the next level of production, if that's truly the case and it's supported by scientific research. So I'm going to start with the early lactation disease, and we know that morbidity is a problem of early lactation. So if you look at diseases that are very common, one of them being metritis, so the cow that has uterine discharge that induces an inflammatory response because of the damage that's taking place. You can see that metritis is a disease that happens in primarily the first 2 weeks, with the median days to diagnosis somewhere here between 6 and 7 days postpartum. But then if we look at the disease that affect tissues other than the reproductive tract, so what I labeled here non-uterine disease, we can see that most of them, the initial diagnosis takes place here in the first 3 weeks postpartum, such that if I overlay the uterine disease with the non-uterine disease, to no surprise, what we see is that about 1/3 of the dairy cows will be diagnosed with a clinical disease in the first 3 weeks postpartum. And of the clinical disease that are diagnosed in dairy cows, almost or a little over 3/4 of that, the initial diagnosis occur in the first 3 weeks of lactation. Okay. The problem with this is that anything that happens in early lactation, good or bad, typically will have a legacy effect or impact in the remainder of the lactation. So obviously disease is not something that's desirable, and it does have negative consequences immediately, but many of these diseases will have negative consequences later on. One of the immediate consequences that disease induces sickness behavior that is typically expressed by reduced appetite. So anorexia is a common find, and it, to no surprise, in dairy cows is no different. They basically eat less. But the interesting aspects that disease can also alter nutrient partitioning in animals. And I'm going to use this example here from beef cattle, this really cool experiment in which they induce disease, and in this particular case, they induce respiratory disease just because that's the disease of interest for a lot of people in the beef world. So they had a treatment group in which they remain as control animals. So these steers here receive saline intratracheally. So the idea here was to have a sham treatment, but not induce any inflammatory response. And then they had a challenge group in which they infuse intratracheally a particular amount of Mannheimia hemolytica, which is a bacteria commonly associated with respiratory problems in cattle. Okay. The cool thing about this experiment is that these animals were multi-catheterized, and by having catheter in key blood vessels in the splenic tissue and the splenic bed, they are able to quantify not only concentrations of different analytes, but by using a marker, they can quantify blood flow and therefore look at flux of nutrients across the portal drain viscera, the liver, or the entire splenic tissue of those animals. And so these catheters come out here in the dorsal part of the animal, and there is continuous blood sampling to then quantify blood flow and venous arterial difference in those animals. Okay. So the animals they were using this particular experiment were these Angus steers that have inflammatory response because of pneumonia that was induced through the challenge. So I want to make a parallel of this disease with a common disease that affects anywhere from 15 to 30% of the postpartum dairy cows, which is puerperal metritis, often associated with systemic signs of inflammatory response, such as increase in cytokine concentrations in blood, increase in acute phase proteins in blood. So there's a lot of similarities, although the two diseases, they affect different tissues. In terms of inflammatory response, they have some commonalities. Okay. So they measure lots of things, one of which was the flux of amino acids across the liver. So here we have in the Y scale the amino acid flux in millimoles per hour. So whenever the bars here are below 0, it means that that particular tissue, in this case the liver, is extracting or using more of that amino acid or group of amino acids than it is secreting. In other words, through the arterial supply from the hepatic artery and through the venous supply from the portal vein, there will be the uptake of nutrients. And the outflow of nutrients would leave the liver through the hepatic vein. So if the flux of nutrients going to the liver is greater than the flux of nutrients leaving the liver, it means that the liver is using more than it's secreting. The value would be negative. And to no surprise, for the essential amino acids, both treatments have a negative flux of those amino acids because the tissues are unable to synthesize them because those tissues don't express the specific enzymes for those amino acids. But we can see here that the challenge group, which is the red bar, has a more negative value than the blue group, which is the control. So inflammatory response increases the uptake of amino acids by the liver and increases the utilization of those amino acids by the hepatic tissue, resulting in a more negative net flux. Now, if we look at the non-essential amino acids, those that can be synthesized by mammalian cells, we see now that the control group or the control treatment has a positive net flux, whereas the challenge animals, they continue to have a negative net flux. When they overlay the two and they looked at the total amino acid flux, we see that the differential is quite larger. So I calculated this using the average molecular weight of the 20 proteinogenic amino acids, and this results in approximately 2.6 moles per day based on this flux. And based on the average molecular weight, this would result in about 380 grams of amino acids for this 400 kilogram steer. If we translate this into a lactating cow at a 67% efficiency of use of amino acids extracted to be incorporated into milk protein, this would be the protein equivalent of 8 kilograms of milk. So in other words, when animals develop disease, to no surprise, performance is impaired. One aspect is because intake is depressed, which is what we typically think, but also because the use of nutrients now is partitioned away from the mammary gland. And the idea here is that animals are trying to contain an infection or inflammatory response. Okay. So the second aspect that was brought up by the nutritionists was that dairy cows, to have greater production, will require better quality forages. Okay, so I just to illustrate this point in an easy manner, I have two diets here, okay, that would be fed to a group of high producing dairy cows. Cows producing 48 kilograms of milk with 3.7% butter fat and 3.1% protein. And I'm illustrating here a diet that will provide to those cows approximately 69 megacalories of metabolizable energy and 3 kilograms of metabolized protein, given that these cows would eat 24 kilograms of dry matter. But this initial diet has moderate quality forages, basically corn silage and alfalfa silage with a high NDF content and with an NDF that has a high lignin content. So each of those squares here represent about a kilogram, and each of those rectangles a kilogram, and each of the squares represent about half a kilogram. So in this 24 kilograms of a diet, in order to meet the needs of this cow, I am restricted on how much of this moderate quality forages I can feed, such that the inclusion in this particular case to get to the numbers that I desire to allow this level of production, I was restricted to about 42% of the diet dry matter. Okay. On the other hand, if forage quality is improved, now I have two extra rectangles that replace corn grain in my diet that would still allow me to formulate a diet to supply the same amount of energy in terms of metabolizable energy and the same amount of metabolizable protein, but now allowing me to increase the inclusion of forage in the diet from 41 to about 50% and still meet the needs of this high producing cow with the same amount of dry matter. So I guess let's see if this is really true. Through experimentation, people have demonstrated that. So let's look at this experiment from Cornell University in which they manipulate the quality of forage by feeding either conventional corn silage or brown meat rib corn silage. The brown meat rib corn silage would be the corn silage with less content of lignin and therefore more digestible fiber. And this should allow cows to consume more feed. And not only they would consume more feed, but hopefully the feed would be more digestible and therefore they would extract more nutrients. Treatments were applied from 3 weeks before calving to 3 weeks after calving, right at the transition period, okay? After 3 weeks postpartum, cows were fed exactly the same forage. So this is the dry matter intake response during the first 3 weeks in lactation. And we can see that cows that were fed the higher quality forage consume about 2 kilograms more dry matter in the fresh period. Once they were moved to a common diet, dry matter intake was exactly the same, about 25 kilograms of dry matter, which we can see here in the figure. But look at what happened to fat-corrected milk. During the treatment period, the first 3 weeks postpartum, these cows that were fed higher quality forage with less NDF, less lignin, they produce about 4 kilograms more fat-corrected milk. The same was true past the period of treatment. So from weeks 4 to 15, so from about 22 days in milk all the way to 105 days in milk, despite of the fact that cows were now fed exactly the same diet, those that were initially fed higher quality forage were still producing another 2.6, 2.7 kilograms more milk for the remainder of the experiment. So it's not uncommon for a lot of the things that we do, either good or bad, very early in lactation, they tend to have a legacy effect past the period of treatment. So if it is something bad, that would have ramifications later on that are also bad. If it is something good, like in this case, they will have positive ramifications later on past the period of intervention. So this is demonstrating that, yes, forage quality is important. So now if we look at the similar approach from a crossover design from Masahito Oba and Mike Allen at Michigan State University, they look at the change in milk as cows move from a conventional corn silage to a brown meat rib corn silage. So from a lower quality forage to a higher quality forage, according to the level of production of those cows. And what they observed was this slope here, indicating that as they change corn silage, there was a positive change in milk because we see most of the cows are above the 0 line here. But the rate of change or the increase in milk yield was greater for cows that initially, before treatments were imposed, had greater milk yield. So this is telling me that higher producing cows respond to a greater extent to better quality forage than lower producing cows. And this makes sense because these cows that are producing 50 kilograms of milk, they have to consume more total dry matter. And to consume more total dry matter, they either have to digest feed faster in the rumen or they have to pass feed faster in the rumen. And by providing better quality forage, probably both aspects, digestion rate and passage, will be enhanced, allowing the cow to consume more kilograms of dry matter and for each kilogram to extract more nutrients. Okay, so it seems that the nutritionists were right again, that this is an important aspect. Remember, the third one that they thought was important is dry matter intake in early lactation. And I want to go back to what Kent already mentioned, this aspect of selecting cows for improved feed efficiency or for negative residual feed intake. Okay, usually the selection takes place in past early lactation. So a lot of the data that's being generated in the US and other countries are with cows past 50 or 60 days in milk when they are no longer losing body weight. So we asked this question in this data set of about 400 cows, and this is our phenotypic relationships, not genetic relationships. We asked the question whether cows that are more efficient in mid-lactation, are they also more efficient in very early lactation? So mid-lactation here is after 9 weeks postpartum. Early lactation here is in the first 5 weeks postpartum. And in fact, there is a positive relationship. So as we can see, the more efficient cows, the ones that have a negative residual feed intake value, they are also more efficient in early lactation, whether we look at his residual feed intake or residual nitrogen intake. So in fact, the cows that eat less in mid-lactation to produce the same amount of milk, they also eat less in early lactation to produce that same amount of milk. Okay, and the phenotypic correlation is about 0.46. Okay, so it's not extremely high, but it's not shy. We don't know whether that's going to hold true when the geneticists work with the genetic correlation. Okay. So if we look at the two extremes in that population of 400 cows, the ones that were efficient in mid-lactation and the ones that were not efficient in mid-lactation, what happened to them in early lactation, in the first 5 weeks postpartum? So as expected, the ones that were more efficient, they also ate less in early lactation compared to the ones that were less efficient in mid-lactation. Okay. Their nitrogen efficiency was greater, so about 31% of the nitrogen consumed was converted into milk nitrogen, whereas here it's only 27%. But the point that I want to make here is whether we change body energy content. So this is the change in energy content. In this case, they are mobilizing body tissue based on daily changes of body weight, and we calculate that energy content based on the body condition score of the cows, because one of the concerns would be that increased efficiency in those cows would result in animals perhaps losing a little bit more weight. As we select based on what we observe in mid-lactation, maybe those cows will lose a little bit more weight in early lactation. And what we found, in fact, that there is a slight increase in energy loss from body tissues, but not a huge difference. Okay, and the cows had exactly the same milk production. So in fact, selecting for greater feed efficiency in mid-lactation seems to also enhance feed efficiency in early lactation. There is a small energy cost in terms of body reserves to those animals. Now, because we are reducing feed intake in early lactation, are we compromising total lactation milk yield? And what we observe in this data set, this is our phenotypic data, okay, is that there is almost no relationship. So you can see here that the R-square is only 2% or less, and this is dry matter intake in the first week with 305-day milk yield observed in those cows, and we have primiparous and multiparous cows here in this data set. So you can see that the slope of that line after adjusting for parity and other aspects is only 80 kilograms, meaning that there's only a small increase in milk yield if cows were to eat more in early lactation based on this association, or eat more in the second week of lactation. Okay. Now, is there a difference in milk yield at 305 days, depending upon the RFI of cows in early or mid lactation? And we see no relationship between the two, meaning that in fact, as we select for improved feed efficiency in those cows, we should not see any loss in milk yield in the entire lactation, despite of the fact that we're basing our phenotypes in a restricted period in the entire lactation. Okay. So I just want to move on into two other aspects that nutritionists did not, they might have identified, but I asked them to select only three. The first one is supply of metabolizable protein. to cows in early lactation, because there's a lot of debate of reducing protein in diets of lactating dairy cows. And I want to use this experiment here from Ohio State University. They initially enrolled 63 cows, but only 56 contribute to the analysis of the data. And they had 3 treatments. These cows were fed from 2 days up to 23 days in milk. So a period of 21 or 22 days postpartum. A diet with 16% good protein, what they call a control, or a high metabolizable protein diet in which they increase the crude protein content of the diet by manipulating some of the ingredients. And in this particular case, they use a little bit more heated treated soybean meal, and they also use corn gluten meal in their diet. So they increase about 2 percentage points of crude protein, but they didn't make major improvements in amino acid profile. But obviously, the total amount of amino acids per kilogram of diet is going to be more. Or they increased the amount of crude protein, not as high as in the high MP here, but they improve amino acid profile of the diet. Okay. So let's just look at how these animals, these cows, responded. They had multiparous and primiparous cows. So there was no difference in dry matter intake in the first 3 weeks of lactation or in the first 23 days in milk. But because these 2 diets provide more MP, these cows consume more MP based on NRC predictions. Okay, just because each kilogram contained more MP. Consequently, the MP balance for the first 3 weeks postpartum was less negative in the diets that provide more metabolizable protein. And the cows responded. They increased milk production about 2 kilograms in both diets, either in the one that had greater crude protein, but not as superior amino acid profile, or the one that had a little bit less good protein, but improved amino acid profile. So we got to be careful with early lactation cows restricting the supply of protein. And I want to show you this data here from Denmark that is a quite interesting experiment. And what they did here, as opposed to allowing the cows to eat, they basically fed exactly the same diet postpartum to these cows, okay? A diet that contained 16.4% crude protein, but they infused abomazally a mixture of amino acids that resemble casein amino acid profile. And the product that they infused was about 88% metabolizable. And they had a stair-step infusion rate. So for the first week, they infused about 700 grams of metabolizable protein coming from this amino acid mixture, and then slowly decrease. They decreased 28 grams per day until 500 grams per day, and then again they went through 3 days of this plateau, and then they decreased again until 30 days postpartum. These animals had rumen cannulas. They were multi-catheterized, and the infusion was done in the abomasum. I just want to show you what happened in terms of animal response in those cows. By providing more MP, cows did not eat more. So they ate exactly the same amount of dry matter, but as you can expect, the total net energy consumed, the total metabolizable protein consumed was substantially more because of the energy provided by the supplement and obviously by the amino acids provided by the supplement. And you can see that these cows responded substantially, about a 6 kilogram increase in milk yield in the first 30 days of lactation. So this is telling me that if we are able to make cows to consume more metabolizable protein, they are eager to respond in terms of milk production in this very early lactation. The mammary gland is sucking up the amino acids that are flowing through the bloodstream at that stage. Just the last 2 things, can we alter tissue response based on the fats that we feed? Okay, and this is an experiment from Michigan State University from Adam Locke's group, in which they fed very early lactation cows a controlled diet without any supplemental fat, only the fatty acids from the basal ingredient, or they supplement the diets with 1.5% fatty acids with different ratios of palmitic to oleic acid, 80 to 10, all the way up to 6 to 30. Okay, and we can see that feeding fat did not increase dry matter intake or did not change dry matter intake in early lactation, which is the comparison between the black line with the average of the three colored lines. But these cows responded in milk yield substantially. So energy corrected milk increased about 4 kilograms per day. Some of the increase is because of the increase in milk fat coming from the efficient transfer of fatty acids into the mammary gland for milk fat synthesis, but some also came from body weight losses, particularly in the diet with high palmitic acid. So much so that when they measure NIFA and insulin concentrations, the cows that were fed the higher content of palmitic acid in the fat supplement, they had higher non-sterified fatty acids, suggesting increased lipolysis, perhaps because they had lower concentrations of insulin that usually would have anti-lipolytic effects. But one thing that we need to think about as we move cows into higher production is that fatty acids have the ability to modulate some of the hormonal responses, one of which is the ability of palmitic acid, which we often feed in diets because of the positive effects on milk synthesis, to alter tissue sensitivity to insulin through the synthesis of ceramides. So palmitic acid is used for de novo synthesis of ceramides, and ceramides have the ability to block insulin transduction cascade. So when cells are exposed to ceramides, this suppresses phosphorylation of protein kinase B, also called AKT. And this protein here is part of the transduction mechanism of insulin. And basically what this does is reduces insulin sensitivity or increases insulin resistance in the tissue. Therefore, if this happens in the adipose tissue or in the muscle, there will be less lipogenesis, more lipolysis. But this also happens in other tissues. And the example that I'm going to pose here is from reproductive cells or granulosa cells from the follicle. In this particular case here are from humans. So we know that insulin should stimulate phosphorylation of AKT or protein kinase B. And that is part of the mechanism of action of insulin. So this is quantification of protein abundance of phosphorylated AKT relative to unphosphorylated AKT. So here we see that by adding insulin, there is a massive upregulation in the abundance of protein in the granulosa cells. Okay, this is true anytime we look in this figure or in this figure, but look at the attenuation of the response as the concentration of palmitic acid in the culture media increase. So this is showing that palmitate can suppress the ability of insulin to induce lipogenesis or suppress lipolysis. So perhaps there will be ways to modulate the fatty acids that we feed as we deal with higher producing cows to allow them to be more or less lipolytic, depending upon the stage of lactation that we desire. Lastly, I just want to point this figure here from a review article from Alex back in Spain, in which he looked at a summary of 51 experiments that reported energy consumption and energy secretion in milk, and he plotted the energy consumed above maintenance of those cows, accounting for changes in body weight and body energy, okay, relative to the amount of milk energy secreted. So in theory, if we are dealing with net energy for lactation and we accounted for by the needs of maintenance of the cow, as well as the changes in body weight gain or loss, everything else should be diverted into milk energy, okay? And that would be true if the animals responded in the dashed or the dotted line here, the theoretical line of one-to-one efficiency. But we can see that that's not the case of this data set. And the point here is that it seems that as animals become more productive or they consume more calories, in this case here, these cows are consuming 35 megacalories above the maintenance needs, which would be the equivalent of about 50 kilograms of milk. But on average, they are only secreting about 30 megacalories of energy in milk, the equivalent of about 42 or 43 kilograms of milk with 3.5% fat. So this is telling us that as production increase, perhaps the efficiency of use of energy in the diet to be converted into energy in milk will be reduced, either because maintenance needs of the cow changes or other aspects. So just to summarize, forage quality will become more critical as production increase. That allows for high forage diets to be fed, which is probably healthier and probably cheaper. We need to implement diets and health programs to reduce the risk of disease. Disease suppress appetite, alters how nutrients are used. The animal shifts its priority from production to survival. Preliminary, very preliminary data, it's all phenotypic, suggests that as we select for improved feed efficiency, early lactation energy status should not be compromised. Okay, at least that's what we can say at this point. I think early lactation cows, we need to be cautious about limiting protein, and in fact, we should probably supplement greater amounts or greater concentration of MP than we probably would typically think about if we want to optimize milk yield. I think in the future, we need to refine fatty acid feeding for different stages of lactation, and perhaps we're going to feed things that promote less lipolysis very early on, and then we're going to target fatty acids that are important for milk fat synthesis, tissue response, and maybe other aspects. Lastly, the efficiency of net energy for lactation used for net energy synthesis in milk seems to decrease as production increases. And perhaps we need to rework the energetics, particularly the maintenance requirements of a cow as we move into herds with very high producing animals. So thank you very much for your attention, and I guess Brian will take over from now.
Thank you so much, Jose, and again, thank you to you both for the excellent presentations, which has set up very well the discussion session. Still time for people to send their questions in as we go into this discussion session. I think Jose and Kent can see the questions, so I might ask some, but if you want to also just jump in and address some of the questions directly, please do so. So just our first question that I really liked, a very practical one. So better feeds or quality forage in the farm should be fed to fresh cows.
So can you say it again, Brian?
Yeah, right. So you showed in one of your slides that the BMR slides, how much you improved. And so if you had to make the choice, would you feed the BMR high quality silage to your fresh cows?
Unquestionable, yes. So you should feed your best quality forage to your early lactation high groups. It's unquestionable that. You know, I think with the idea that we need to limit energy intake pre-part, and a lot of times people have moved the opposite direction. We became accustomed to add very low quality, horrible forage like straw to cow diets. But in early lactation, I think we need to use the thermometer of how much I can feed of a better quality forage without causing any digestive problems. And the way of doing that is, if inventory allows, you just need to feed more forage in the diet. Yeah, I think it's unquestionable. If I have to choose between two piles of corn silage, one with 3% lignin and 38% NDF, and the next one with 45% NDF and 3.8% lignin, the first one will go to the fresh cows and the high pens, and the other one will go to the low pens.
So one for mostly for Kent, but by using genomics and heavy selection of specific traits, are we heading towards genetic lines like it happens with poultry or swine?
You know, we could certainly go that route. I mean, there have been a couple of the studs that have looked at that a little bit, just mostly in terms of how do you help the farmers control inbreeding and sort of have, you know, maybe 3 lines that don't overlap so much that you can just interbreed, you know, the red line and the green line and whatever, right? You know, very simple mating strategies. But I mean, in reality, our breeds are selection lines, right? I mean, Jerseys, Holsteins are selection lines. They just evolve, you know, they've been selected over centuries, not, you know, not short period of time. But we could go that route. I don't know. I mean, there's so much variation within the breed still, that you can kind of find what you want anyway. You don't necessarily have to have a fertility line and a you know, a milk volume line and a components line or whatever, right? I mean, it's a little harder to do that in cattle too, because of the longer generation interval, though not as long as it used to be, right? But those selection lines work better when you're doing you know, 3 breed crosses, that sort of thing, right? The maternal line mated to the meat quality line and growth, that sort of thing. We may see it, but it's not imminent, I guess.
Actually,
you know,
Jose had a question. I don't know if
Yeah,
someone
I have
had
2 questions
Yeah,
actually.
go ahead. Yeah.
So you showed that slide that you have different species that, as we select for residual feed intake, animals become leaner. I'm assuming in the cattle is from beef cattle data.
Sure.
Is there any initial data from dairy cattle that we know what the genetic correlations are between RFI or feed saved and daughter fertility?
Yeah, there are, and I'm not recalling the exact numbers, Jose. I should. I think Paul Van Raden has done that. If I remember correctly, and it's bad to guess, but I think it was pretty low. You know, confidence range including 0. You know, so I think that's always something we're gonna have to keep an eye on, right? I mean, the last thing we want to do is select for skinny cows and more health problems. And I think we have to be really more careful in selecting for feed efficiency in dairy cattle than we do in meat animals, right? Because more things can go wrong, you know, and we're asking these animals to do more different things. So I think that you know, that's something that we have to monitor, you know, pretty darn closely. But it wasn't alarming to me when I saw it. Now I can't remember now the exact number. It was pretty small.
Okay. So the other question is, with genomics, can we make sense and disentangle?
Yeah.
You know, the what do you call it?
The recessives, right? Yeah, the genetic defects.
Well, the genetic, the inbreeding. I'm assuming a lot of the negative effects of inbreeding is coming from accumulation or homozygous and accumulation of bad genes, yeah?
Yep.
Or maybe some of the haplotypes that show up. So can we sort that out such that we have daughters of cows and bulls that are relatives, but we don't have the negative effects? Because everybody's going for the same bulls, yeah?
Yeah, no, and the population is very closely related. It's not as bad as dogs, but it's pretty closely related, right? Very, and yes, we can do that. So sort of 2 thoughts here, and we had a student now some years ago. She's at University of Pennsylvania now, not working in livestock, but she looked at, in the Jersey breed, she traced the inbreeding depression back to founder sires, and you know, found that it was different in certain families, which is what you'd expect. What's the genetic load that this old famous bull carries as compared with another one in terms of recessives? And then if you inbreed one animal's daughters, one, you know, famous you know, foundation bulls, you know, future offspring, great offspring, you have more genetic cost of, or more cost of inbreeding than others. And so we know that, right? So we can identify that. But the other part that's interesting is the folks at USDA, particularly Paul Van Raden, who I mentioned earlier, you know, figured out that just by simple math, you can look at regions of the genome where there are missing homozygotes, right? And so those are lethals, right? I mean, not every homozygosity at every undesirable allele results in lethality, right? Sometimes it's just reduced performance, but that was really clever. And so the cost of inbreeding has gone down, right? Because we can identify these genetic defects so fast now and breed around them or breed them out of the population, right? So we have more tools. I mean, the inbreeding is a tough thing, right? Because we can get rid of it instantly by just stopping selection, right? And let everybody be a parent and have random mating and go back to the good old days of 100 years ago or whatever, right? But we're not going to do that because we want to make progress, and so we have to have this balance. And so there's a lot of hand wringing that happens about inbreeding, but not actual action that happens, right? So we worry about it, but we don't really do anything because we want improved performance more than we're worried about inbreeding, if that makes sense.
It's hard to go away from those top bulls, yeah.
Yeah, there are, but it certainly makes it easier to manage it now with genomics because we just know so much about the genome. There was a question, Brian, you said we could kind of look at some of these, and I can't remember now exactly what it was about the NRC, and I'd be curious to hear Jose's thoughts on this. And I don't want to pick on it because it's a nutrition Bible, right? And I'm a geneticist, so that would be kind of bad for me to say nasty things about the NRC. But I think from an outsider's viewpoint, and we look at this topic, I think we're beyond the point now where a once every 20-year Bible on nutrition requirements is a good thing, right? I think we're making such rapid genetic change, we're making such rapid changes in management and nutrition. There's another question about rumen microbiome. That wasn't a thing in 2001, right? Not too much. Maybe a few people were working in that area. Now there's a ton of work. And so I think we need, and I think you guys recognize this in the nutrition community, a system that can be updated more rapidly. And it's not just that it's published every 20 years, let's say, but in 2001, that was the end product of several years of discussions about published literature that was published, the most recent of which would have been in the late 1990s, which would have been experiments done in the mid 1990s and earlier. So really, it's not even 20 years. It's probably 30-year-old data right now, right? The actual trials that went into that. So we need sort of a more robust and interactive system where you can adjust it on the fly, I think, but that's just my view from the outside. Jose, I don't know what your thoughts are there.
Yeah, no, you're right. Yeah. So you have to have data to substantiate the recommendations. So there's two aspects, is that obviously there is a delay, the first one, but the second one that is more eye-opening is that the fundamental research that is lacking from a previous cycle is never addressed in the next cycle.
Right.
So
right.
A lot of times the data that provides the backbone to generate equations for nutrient needs, they are from fundamental research that was performed in the 60s or 50s or 70s. Best example is mineral nutrition. All the isotope work was done in the 50s and 60s. Nobody does that now. Therefore, we do trial and error type of experiments. Yeah, we add more, we add less, and then we look at response. It's not mechanistic to a great extent. A lot of the experiments are not mechanistic.
Why is that?
Because of funding. It's because of money.
I mean, those are expensive experiments, right? To be really intensive. And you mentioned yesterday, I think, or the other day in our discussion about, these multi-lactation trials or even a full lactation trial. It's really not a thing anymore, right? We just are doing 6 weeks, 8 weeks, 12 weeks at a time, and that's it.
That's right. So to use stable isotopes, I'll give an example. We're looking into calcium stable isotopes per animal just to have the enough amount of the isotopes, just because of the size of the cow. It costs about $12,000 to $15,000 per cow just to do an experiment with one animal. Now, if we were to infuse radioactive isotopes, then we have to tank the cow. So there's a lot of fundamental questions that are never answered because it's expensive to do in cows. That's one aspect. The second one,
so coming to that point, Jose, sorry to interrupt, but as part of the you see some of that early work done by USDA scientists and others where you had a sort of a stable research budget, maybe not the sort of pressure to publish 15 papers a year. You could publish one really good one and spend a ton of money doing it, and people were happy, whereas now that will get you fired or not tenured at least. Is that an issue?
It's a big issue. I think it's a big issue. Yeah. And the other aspect, obviously, in the 60s and 70s, or maybe the 70s and 80s, university research were the lead places in terms of progress or where things are going to move in terms of infrastructure. Today, university research farms they lag behind. So we go to commercial farms to see what they are doing, and then we can emulate that when we build the next facility. And very few people revamp their facilities. So the gap between what University of Florida herd does and what the best herds do is huge, and used to be narrower or used to be close. So how many university herds have 30,000 pounds of milk? Michigan State and University of Wisconsin, and maybe somewhere else in Europe. Everybody else is in the 24 to 26,000 pounds, whereas we're talking about the cows of the next 10 years, there will be probably many herds with 32,000 pounds or 15, 16,000 kilograms per cow with 1,000 cows, 2,000 cows. Yeah, it is hard to answer those questions because a lot of those fundamental questions are done not with the ideal cow. It's not an early lactation cow. A lot of the fundamental physiology is done with a cannulated cow, with a multi-catheterized cow that oftentimes is only producing 35 kilos of milk. So, and funding is an issue.
Hey guys, I'm going to jump in here because your discussion is provoking quite a few additional questions. So I thought we'd go to some of them. Okay. So one here is, what genetic number or numbers are available to help determine quickly what the potential of a herd is? Is this herd capable, i.e., is this herd capable of 150 pounds average?
That should be a simple question. It's really not, because we can estimate the genetic potential, but the management potential of the herd varies greatly. So what I was trying to illustrate with that scatter plot was really the differences between animals. We have big herds now, we have big pens, and you can look at a pen, or you can look at all the first lactation cows at a Lions dairy or somewhere, right? And you could say, wow, are you getting the potential? Which means, are the best of those animals exceeding the genetically poorer animals by as much or more than we'd expect? That's kind of what you can do as a diagnostic. It's hard to say you should be getting 97 pounds or 112 pounds, because that then really gets into mostly non-genetic questions of where is kind of the average. What we can differentiate is what's that slope look like by genetics, and are you capturing the extra, if that makes sense. Maybe Jose can answer that more eloquently.
Yeah, I was going to ask Jose, from a nutritionist point of view, do you think we're 10 pounds or 100 pounds behind nutritionally the genetic potential of a cow?
It's a good question. I don't know, because
are you behind at all? Maybe we're not behind at all.
That's why I didn't become a geneticist, because I wanted to do the difficult job of being a nutritionist. Because I thought we were always behind. So.
The issue is how to disentangle this. Yeah, you can compare different farms. Yeah, I can probably go and genotype animals or get the genotypes they have and look at their breeding values genetically and look at how they perform in farm A, B, or C, or D. And I could attribute that to everything that farm does. How do we know that that is all nutrition? Yeah, there's lots of things that influence production. So it's hard to pinpoint a single one. So if I were to think about the potential of the breed or the cow, the one that I know, phenotypically, the highest producing cow has produced 36,000 kilograms of milk, and our average cow in the US produces 11,000 kilos, so we are only 30% of the potential for the maximum production per cow. Genetically, I'm not sure what's the top cow in the country. I really don't know. So yeah, I don't have an answer for that.
So yeah, so that was a great question, because if you don't have answers, it must be a good question, right? You know,
Well, it's a tough one to answer. It's to know how far are we from allowing that cow to reach the potential. There's so many factors that influence that. So
here's another one from our friend Lucchini, Daniel Lucchini. So if selection aims to have more resilient cows and not only produce more, but also being healthier, will it be more profitable to capture the milk potential on a full third or even a fourth lactation of the cows, considering that it takes 2 lactations to break even?
Yeah, Daniel's still trying to get me on that last slide because he really hates that. No, no, I'm joking, but sure, right? If you look at individual cow level, you say, when do you pay off your bills? It makes no sense, right? If you look at the whole, and maybe it makes no sense at all, right? But if you have to look at the whole system, right? And if you said, okay, now I capture more value in the beef enterprise, or now I spend less money on my breeding program, you know, so therefore I also spend less money on labor. That's kind of the kind of thinking I was trying to generate. And the cow would have to be different, right? We would have to have a cow that doesn't ramp up to her peak yield over several lactations. We would want a cow that matures more quickly, maybe calves at 1.5 years and milks at 90% of mature value right away, which we could do by selection. It would take a little while, right? But we couldn't just take today's cow and today's management and just make a change like that, right? I mean, we'd have to change the whole system if we even thought it was a good idea, which it may not be, right? But at least it got Daniel thinking, right?
Right. So it's another Daniel type question, but it's me that's asking it in terms of, from a sustainability point of view, if we think of the whole farm system, and probably in the future, or even in certain countries like the Netherlands already, where the nitrogen efficiency part is really important, because basically they have severe limitations now on what they can do and apply to the limited land space they have. Maybe there's there's another element to this efficiency or sustainability we need to take into account when we're thinking about the type of cow we want to breed for the future.
We stayed away from nitrogen efficiency, and again, Jose's the expert here, not me. But when this first project started, 9-10 years ago on a feed efficiency, we stayed away from it with the logic that people were overfeeding protein, and so how do you evaluate the efficiency of something that's being fed at higher levels than it's needed, right? But the question maybe I would, before I turn it over to Dr. Santos, is can we now, with the genetic tools we have, identify those animals that are likely to lose production if we reduce the protein, and those that aren't, right? Because if you overfeed protein just in case, then in case some cow drops, well, then if we could figure out which cows are, then maybe some cows are happy at a lower level, right? And we could identify that with a DNA test. I don't know.
Well, just by selection for residual feed intake, automatically you're going to be more nitrogen efficient, yeah?
Yeah.
By doing that, because the cow is, unless we are producing milk with less protein, but that's not the case, because the protein is part of the equation calculating energy corrected milk that's being produced by the cow. But just by eating less dry matter sheets, less total nitrogen, therefore nitrogen efficiency will go up. So we will make some progress on nitrogen efficiency. So we'll go back to what Brian mentioned. For countries that have restrictions on how much nutrient they can dispose of in their land, this will be a plus in terms of value added to the selection program.
I just wondered, another idea out in left field is perhaps we could select for cows who can recycle nitrogen better, so I just wonder,
broadly in this area of nutrition interaction with management, it makes me think about, like the animal breeding community versus plant breeding, and I'm not an expert on plant breeding by any stretch. The whole tetraploids and stuff kind of makes my head hurt, and I don't really understand it. But I do know that you have hundreds and hundreds of plant breeders out there, because you have to test every variety and every soil type and moisture level and so on. And genotype environment interaction is a huge thing that they focus on understanding. We in animal breeding, especially in dairy cattle breeding, have treated it as noise. And we've said, we want to know the genetic potential, and everything that nutritionist does, veterinarian does, that's noise, and we want to get rid of it. And so we can focus only on what's the average genetic potential of this bull's daughters. And that made sense in a progeny testing system, because you were averaging the performance of that bull's daughter out over hundreds of herds, right? Now with genomics, we got to flip it all around, right? So now we can know about the individual female's genetic potential at great detail. Now we can know more, because herds are bigger. We can know more about their management, their feed quality. We have the technologies and the data streams to do that. You couldn't do that with 1,000 little herds, but with a smaller number of big herds, you can look at G by E interaction really effectively. And I think we still are trying to get our heads around whether the work that veterinarians and nutritionists and others do is a feature or a bug, right? We treated it as a bug in the past, and now I think it's a feature, right? And we got to get our heads around how to link those two together.
Yeah.
Well, guys, I think I know that Jose, I don't know if he needs to fill his glass up again, or he still has some of his milk left. But
I still do.
let's
See,
say
it's wine and cheese.
so let's
Nice.
have one last question. Say about again, are some cows more efficient because of, let's say, the size of their viscera, i.e. the size of the rumen they have, etc.? What's your feelings on, let's say, organ size, or at least digestive organ sizes, as being part of selecting or cows being more efficient?
That sounds like a physiology question. I'm out.
Well, my intuition would say, if we're looking at residual feed intake or feed efficiency, like residual feed intake, I would say that they probably have smaller, at least in beef cattle, larger viscera accounts for greater maintenance requirement. So I would say that if I had to really document that, my hypothesis would be that more efficient animals probably have smaller visceral, smaller intestines, smaller rumen, less omental fat. Maybe completely wrong, but that would be the case. Perhaps more productive cows, irrespective of their efficiency, they probably have larger viscera, because not genetically, but phenotypically, larger animals produce more milk. At least first lactation animals, they produce more milk, and therefore, if they're larger, they probably have larger gastrointestinal tract. Genetically is the opposite. Larger animals, there's a negative genetic correlation, correct, Kent, between body size and milk yield, but phenotypically, I think it's a positive genetic correlation.
It's pretty small, yeah.
But I would say they probably have smaller viscera, so there is less energy used for protein turnover, for accumulation of tissue, and things like that.
Well, again, on behalf of Adiseo and the audience, I would just like to thank you both for, again, an excellent evening and a very stimulating discussion. And I hope next year that we will be back together physically, so that we can obviously not only enjoy whatever topic Daniel Lucchini comes up with for next year, but also be able to enjoy really the wine and cheese. So thank you. One other thing before I go, just to say that this has been recorded and will be available within a week on our feed channel, and everybody who's participated will receive an email indicating when it has been posted. So if there's anything you want to go back and revisit or recommend to a friend or colleague, this will be on our feed channel. So thank you again, and have a great evening. Good night.